One on board.
If it's fun, we'll go. I wasn't a sister. It's not like I.
Is it just like a
Welcome everyone. It's a dramatic weather day. We have a nice little drama show for you today. Welcome to Ada's classmates, colleagues, and friends and teachers. From her formal public defense of an accounting of her time here, both academically but mainly research-wise. And welcome to also her family, her sisters and mom who came in from out of town, and Dad who's joining us via Skype. I'm Phil Hanna, I'm Ada's PhD thesis advisor, examiners. And it's my pleasure to tell you recaps before she begins her scientific talk. So Ada was a scholar at East Tennessee State University. She did undergraduate work there, and she did her master's degree there. And she was while she was there, she fell in love with microbiology. I'll tell you about that. But what you may not know is that she was also an athlete, and with a specialty in violent athletics. And she won lots of awards and fellowships there on the academic side, not on the athletic side. And no, maybe she did. And she worked in bacteriology. And as a matter of fact, she worked in iron acquisition, something she worked on with us. And as an undergrad, she did MRSA, iron acquisition systems. And then she did iron siderophore transport systems in E. coli for her master's degree work. And she came to us in 2012 and joined my research group the following year, and she kept on with that. And iron acquisition in pathogens and pathogenic bacteria is a mature field. You're going to find, if you follow this field at all, that Ada's work, not only the anthrax iron acquisition systems field forward, but also focused on aspects of iron acquisition that aren't really covered yet in any of the pathogens, very few. And she's been pushing that border ahead in advance of, in my opinion, much of the rest of the field, and I'm very proud of that. She has some nice publications with that. She's an excellent writer. And she's done a lot of writing. She writes on her own research and her own research topic, but since she's been here, she writes for the departmental newsletter. She's contributed to ASM's monthly magazine, Microbe. She is founder and chief editor of what's it called? Can't remember that. Yes.
Big transition.
That's right. And she's contributed in the classroom. She's an excellent scholar. She's contributed scientifically, which I'm very appreciative for. And she's also served. She went to Ethiopia to teach for a month. And then she was invited back a year later to Addis Ababa to help design the curriculum for the medical school there. And very proud of her for that. She's been a Watkins Fellow, it's an ASM fellowship, MMMP Fellow here at U of M, and also a Rack of Merit Fellow. Presuming success of her public defense and her private exam, Ada will transition from bench work to scientific writing. She will be an American Society for Microbiology postdoctoral fellow working with Dr. Pat Schloss, doing analysis of authors, publications, journals, impact, and critical analysis of the publication system. That should start this month, or as soon as she recovers from her graduate work. The title is of our talk today is Inside or Out, Characterizing Petrobactin Use in Bacillus anthracis. And I invite you to enjoy Ada's talk, critique it, and ask her ask her anything you're interested in that she didn't describe to you. Part of the question, she specifically requested very difficult.
All right. Thank you guys for being here. I am going to start things off a little bit differently. I know that when I go to a defense, I'm like, oh, that was cool, I have a question. And then by the time we get through the acknowledgments, I kind of forgot who it was. So I'm going to give you guys my acknowledgments up front, and then we'll get into the science and hopefully have a nice discussion at the end. So with that in mind, and Bill's glowing description of my resume, I guess, I'm going to give you an idea of how Phil is. So this kind of says almost everything you need to know about Phil. This is him at Angelo's, went there with Travis and my son, who was sick in the daycare. And Travis was also there with his daughter. And as you can see, he's very supporting, encouraging soul to have been so supportive of me in my journey through grad school and also having a child during grad school. So this is later that day. Phil let me get some lab work done, holding Dax, and within probably 10 minutes we had Dax asleep, which can be a feat. And this tells you something else about Phil, which is that when you need him, he's there, and he helps guide you in the direction that you need to go. Yeah, this is sleep. So there are a lot of people that think research-wise. In particular, my co-grad student in the Hannah Lab, Travis, Uncle Travis. There are a lot of previous members in the Hannah Lab whose work I've built upon. Of course, Paul Dixon, Shandy, and Brian James are all big ones. And then we have number of undergrads who've been really helpful in this. A lot of this work would not have been possible without Ashu from the Sherman Lab. You'll see why. And Zach Mindel in the Swanson Lab has been a really big help lately with some microscopy. Of course, my thesis committee, Carrie, Susie, and now Randy, who is a pinch hit for me when Carol, previous committee member, moved on. So thank you for your suggestions and advice and guidance throughout this process. There are also funding sources, but really they give you more than money. They give you a lot of mentorship and educational opportunity. So for that, I need to thank the NIH and the Department of Training Grants, the American Society for Microbiology, and Rackham Graduate School. There's also friends, all the friends who have made life in Michigan actually enjoyable, despite this polar vortexes. So there have been many adventures from kayaking to Disney-themed birthday parties to adventures in Ethiopia. And thank you, all of you, for being such awesome people. And of course, family. As you know, my dad couldn't be here, but he and my sisters and my mom and my extended family have all been incredibly supportive of my journey. Thank you. Thank you. I'd also be remiss if I did not also acknowledge my undergraduate mentor, Dr. Ranjan Kakarborty, who I hope is on the live stream. And he has been, he taught me a lot about what it is to study research, to be a scientist, and also how to tell stories in science. So I thank him to say that. So with that, let's get on to it. I'm going to give you a couple of different stories about my research in the Hannah Lab. And it's going to be, the first part is talking about how petrobactin gets out of the Bacillus anthracis cell. And then the second portion is going to talk about the role of petrobactin in spore biology. anthracis. The Hannah lab has worked on this pathogen for quite a long time now. It's a gram-positive spore-forming organism. You can see here the sporulation of anthracis. So you can see the phase-bright spores produced by Bacillus anthracis, and these are highly durable, sort of bacterial bunkers. All right. You can also see next to it growth of anthracis on sheets blood auger. Right. So Bacillus anthracis causes the disease anthrax, and anthrax occurs is a disease that can occur in mammals from across the globe, whether it be wood bison in North America, Canada, northern US, or chimpanzees, tropical rainforests, to ruminant livestock and livestock animals. As a disease, anthrax is sometimes also in the news. For instance, the mailing of letters containing anthrax, as well as more recently with concerns that North Korea is developing anthrax as a bioweapon. Once anthrax reaches, once Bacillus anthracis reaches the bloodstream and becomes systemic, it can be rapidly fatal. There are 4 potential routes of exposure to Bacillus anthracis spore, and it's critical that exposure is to the anthracis spore in order for disease to occur. So this can happen either cutaneously, by ingestion of contaminated meats, inhalation of the spores, or more recently by direct injection of the spores by injectional drug users with contaminated product. In order for anthracis to go systemic, the spores have to be taken up by macrophages or other antigen-presenting cells. And instead of being degraded in the lysosome within these macrophages, the spores germinate. And they use toxins, they break out of the lysosome into the macrophage, they replicate within the body of the macrophage, and then bust out of the macrophage into the blood or lymph. And the macrophage is usually transiting through the blood because it's on its way to the nearest lymph node. Once it reaches and begins growth in the blood or lymph, it grows extremely rapidly. There's an explosion of growth up to 10 to the 8 CFUs per mil. Here we have an image of a rabbit arteriole that is almost completely clogged with anthracis bacilli. And this process requires a number of different nutrients. One of the key nutrients, one we're going to talk about a lot today, is iron. Iron is an essential cofactor for cellular proteins, from humans to bacteria and everything in between. Because it is involved, it has a unique ability to transfer electrons. And this redox potential makes it extremely valuable for functionality of enzymes. So in humans, you think of heme, hemoglobin, transferring oxygen between cells. In bacteria, humans, everything in between. Aconitase is an enzyme involved in the tricarboxylic acid cycle. So essential cellular metabolism, enabling growth of the cell. And then hydrogenase is a common enzyme used by bacteria for metabolism of dihydrogen that also requires iron. Even though iron is very useful, everything in moderation. If there is unregulated quantities of iron, that same redox potential that's useful becomes dangerous and deadly because it undergoes something called the Benton reaction that creates superoxide radicals that damage the cell instead of enhancing growth. And so in mammals, there are some dedicated iron-binding proteins that help chaperone iron and ensure that toxicity doesn't become an issue. Within the iron storage within cells, we have ferritin, and iron chaperone between cells, such as in blood plasma, we have transferrin. The activity of these iron-binding proteins serves to reduce the amount of ferric iron available to invading bacterial pathogens to 10 to the negative 18 molar. So that's an extremely tiny amount, and so bacterial pathogens have evolved various ways to scavenge iron during infections. One route is the capture of iron-binding proteins. So some bacteria have proteins at the cell surface that capture a protein like transferrin and steal iron away from it. Some pathogens, Bacillus anthracis is one of them, takes advantage of heme. Heme is sort of an adapter molecule, so the iron can be used as a cofactor. And some capture heme directly from the environment and then gather the iron from it. And nearly all pathogens that require iron use siderophores. Siderophores are small molecules that scavenge iron, and they are synthesized within bacterial cells at extremely low concentrations when the cells are under stress for iron. So there are two different families of biosynthesis genes used for synthesis of siderophores, NRPS and NIS. We'll come back to why that's important in a minute. Synthesis. After synthesis of the siderophore, it gets exported across the cell membrane, where in the extracellular milieu, it scavenges ferric iron. That changes the structure of the siderophore, so it can now be recognized by a dedicated import or dedicated receptor and imported through a dedicated import system. Once inside the cell, the iron is removed from the siderophore for use. This can be a couple of different ways depending on how tightly the iron is bound by the siderophore. If it's bound extremely tightly, you have high affinity, then the siderophore is enzymatically degraded, and then the iron can be reduced and used by the cell for growth. If there's a lower affinity, then an enzyme can simply reduce the iron, and the siderophore can potentially recycle. Siderophores are extremely structurally diverse. There are over 500 different types of siderophores that have been identified. And as you can see, they take on a wide range of forms. Many of the siderophores function using one or two or even both of a couple of iron-binding structures. So, for example, carabactin is considered a hydroxymate siderophore because it uses hydroxymate groups to coordinate a single molecule of iron. Are considered catechol siderophores because they use these catechol groups to help coordinate iron. And then you can have everything in between. Zotobacter is mixed, so it has some hydroxymate, some catechol, and maybe even some other iron-binding ways. Petrobactin is our focus for today. It's considered a mixed siderophore. And Bacillus anthracis can produce both bacillibactin and petrobactin. It has the genes for synthesis of both of these siderophores. Something else I want to point out is that the structural diversity of siderophores means that there is also a wide range of functional diversity. So in addition to the primary goal produced in our eyes of siderophores providing iron, they can also scavenge other types of metals, protect against oxidative stress, and even participate in cell signaling. Petrobactin, even though anthracis can gather heme, produce bacillibactin, petrobactin is the only one that's been shown to be required for growth in vitro and in our mouse models of inhalational anthrax. So on the left-hand panel, I'm showing you growth of Bacillus anthracis in iron-deplete medium. On the y-axis, we have we're tracking cell density, and then over time. So wild type here grows pretty nicely in iron-deplete medium. This is medium that has most of the iron removed. There's just enough remaining to enable growth, assuming a siderophore or other active iron deposition system is present. If we knock out bacillibactin, the growth isn't changed. So it grows just as well as wild type. However, if we knock out petrobactin here in gray, this is the delta ASB strain, then you have a pretty severe growth effect. Looking at mice, when we infect mice intratrachally with spores, we use about 5 times the LD50 dose. Within 5 days wild type mice get sick enough that we have to keep the mice up. However, if you infect mice with spores from the Petrobactin knockout, then they survive throughout the entire experiment. So one reason why Petrobactin might be more relevant for disease than bacillibactin or heme acquisition is that Petrobactin subverts the innate immune response. So bacillibactin is considered a catechol siderophore. And importantly, these catechols are 2,3-dihydroxybenzoate or 2,3-DHB catechol rings. Well, the innate immune system, neutrophils in particular, produce a protein called siderophylin, which has the ability to bind siderophores that have bound iron. And this prevents the ability of that siderophore to return to the cell with its iron payload, so thus starving bacterial cells of iron. Petrobactin, however, instead of having hydroxyl groups in the 2 and 3 locations, like with bacillibactin, has hydroxyl groups in the catechols at the 3 and 4 locations. And what this does is it shifts the structure just enough that it no longer fits into the siderophore binding pocket, and thus is considered a stealth siderophore. So I'm going to tell you what we know thus far about Petrobactin biosynthesis and use in Bacillus anthracis, so that you have a foundation you need to move forward into my work regarding Petrobactin and Bacillus anthracis. Petrobactin biosynthesis is unique in the siderophore world because it involves enzymes from both types of siderophore biosynthesis pathways. There's also a 6th enzyme, ASBF, which is unique to Petrobactin because it is responsible for the generation of these 3,4-DHB. So the structure of Petrobactin are these 2 3,4-DHB catechols bound to 2 spermidine arms with a citrate backbone. Both spermidine and citrate are pretty common cellular metabolites, and so it's the joining and then the 3,4-DHB that are important in Petrobactin synthesis. We know that there's some redundancy in Petrobactin use by Bacillus anthracis. So while we, up until now, didn't understand how Petrobactin was being exported from the cell, work by previous members of Paul and Shandy worked out the import system where there's the receptor FQA, 2 different permeases that can function FQB, FAT-CD, and then there are 3 different ATPases capable of powering those permeases, all to import Petrobactin. And we don't have enough evidence to say one way or another how Petrobactin is dealt with for removing iron for growth. So I'm going to tell you about my work regarding identifying the Petrobactin exporter. To do this, I first had to generate an idea, a list for myself of what the exporter might look like, what it might be, so that I could try to identify it. So to do this, I used a combination of bioinformatics information. On one hand, I went to known siderophore exporters, looked at their protein structure, and looked in the Bacillus anthracis genome for genes encoding structurally similar proteins. And so here I generated a short list of potential export proteins. I also took advantage of a lot of studies that had been done previously in the lab, looking at the transcriptional regulation of anthracis genes under growth in low iron deficiency. So growth in blood, growth in macrophages, and growth in iron-deplete medium. And from here, I looked for transporters that were upregulated under these conditions, because we know that siderophores and their accessory proteins are typically turned on during these low growth conditions, low iron conditions. Right. So this gave me a list of candidate exporters, and then I went and cut each exporter out from the anthracis genome one by one, and screened them by their ability to grow in iron-deplete medium or export Petrobactin. So the hypothesis is that you can't export Petrobactin, you can't scavenge iron, you can't grow in iron-deplete medium. I have a list of 17 putative exporters, and these belong to a wide range of transporter families. The ATP binding cassette family, which uses ATP to power transport, major facilitator superfamily, resistance modulation diffusion family, R&D, multi-drug and toxic compound extrusion, mate family. All of these use proton motive force to facilitate transport. So I made the deletions, and then I screened them for their ability to grow in iron for the medium, and they all grew. So on the left, again, we're looking at cell density over time. We have wild type. You can see it grows quite well. ASB has our growth phenotype. And mutants, I'm only showing you 2 here because all 17 look identical. They grow just as well as wild type. This doesn't necessarily mean that there's an impact on catechol production. There could still be a pretty big hit in the export of petrobactin, but that could be enough to enable growth. So I looked at whether or not petrobactin is being exported from these mutants in putative exporters. I did this using a colorimetric assay, a catechol assay. So it detects any catechols that are present in the supernatant. And here I'm using the presence of catechols as a proxy for the presence of petrobactin in the supernatant. Here's an example. In media alone, there's no color change. Wild type is exporting petrobactin. So you have this nice red-orange color. And then delta ASB, no petrobactin, so there's no color change. And then I can quantitate that with spec. And to show you what this data looks like, on the y-axis we have catechol production, and this is set relative to wild type at 100%. So everything is relative to wild type. And at the bottom, the value given by delta ASB tells us the limit of detection of our assay. Right. So, ready for it? They all look pretty much like wild type. None of the mutants I screened had severe defects in catechol export that I predicted to occur for a petrobactin mutant. And this was a little bit frustrating. I had a lot of strains, put a lot of time into this. I had no phenotype. And I started to think about the assay I was using. And it was focusing in on 3-4 catechol rings. What the assay doesn't tell me is if those rings are actually attached to the petrobactin molecule or not. So I decided to take a step back and look at the entire molecule as opposed to just the catechol rings. To accomplish this, I collaborated with Ashu in Dave Sherman's lab with a new sampling system they had for mass spectrometry. And this is LASY, laser ablation electrospray ionization. The way it works is that you can set pretty much any sample, no processing required. As long as there's water in the sample, you can set it underneath the laser. The laser shoots down, creates a plume of aerosolized droplets. Those droplets have your analytes. So that plume is intercepted by the electrospray ionization to charge it, goes into the mass spec for an output. This is a really powerful technique, in part because the direct sampling without extraction means that you can look for a lot of different molecule types, from lipids to proteins to nucleic acids that might get separated out by separation techniques. And depending on the power of the mass spec, you can detect down to the femtomole level. So I took the supernatants from the mutants that I just showed you and assayed them using LASY. And really just going to show you one piece of data because it's the important one. When we looked at supernatants of one of the mutants, of the 2407, we found that there was an absence of petrobactin, even though there are high amounts of catechol. So on the y-axis, we have intact petrobactin. Again, this is relative percent wild type at 100, ASB at 0. There was, again, no petrobactin in the supernatant of the 2407. When we complement that, adding back a copy of the 2407 onto a plasmid, then we can restore presence of petrobactin in the supernatant. What's even more striking is if we look at the cell pellets. So remember, you can sample anything as long as there's water. So again, intact petrobactin percent wild type. The cell pellet has 400, about fourfold the amount of petrobactin inside the cell of wild type does, which is something I would expect if you can't send it out to the supernatant. If we complement that again, then you restore petrobactin levels in the pellets to wild type levels. So I'm going to tell you a little bit about what we know or predict about 2407, which I have returned Apex for apo-petrobactin exporter, since it exports the non-iron-bound form of petrobactin. This belongs to the R&D family, and the way I found this protein was, incidentally, it has some homology to the mycobactin exporter and Mycobacterium tuberculosis. And the R&D type family has 12 different transmembrane helices, similar to other types of proton low force transporters, and it's predicted to function in a trimer. So you have 3 of them sitting side by side, and they pump solute out almost in a carousel-like fashion, where each protein in the trimer has a molecule at a different point in the export process. The thing about all of this is, though, after I give you my updated model, showing we have our protein exporter now to add to our import system. The thing about all of this is that I'm still kind of hung up on why Bacillus anthracis mutant in the exporter for petrobactin is still growing and still has those catapults and supernatant. So I set out to try to understand how is the iron coming in? My prediction is that petrobactin is either being cleaved, or there's so much petrobactin packed into the cell pellet that biosynthesis gets truncated and it's sending out those fragments. So I wanted to explore this a little bit further to see if we can learn more about the types of fragments that are gathering iron here. I did this in part using a feeding assay. So I would grow 2407 or other strains in IDM, and then at a 6-hour time point, I would filter out the bacteria and take that supernatant and cross-feed it with a complete petrobactin knockout. And then I would look at how these supernatants impacted the growth of delta ASB, presuming that there's petrobactin in those supernatants and these petrobactin components. In some cases, instead of supplementing with spent media, I used precursors for petrobactin, 3,4-DHB, spermidine, or citrate. So everything you're going to see for the next couple of slides, minus one panel, is looking at how these supernatants increase or don't affect the growth of the delta ASB mutant. Okay, so they're all delta ASB mutants. The only thing that's different is what's been supplemented. When we supplement delta ASB with its own supernatant, no enhancement of growth. However, if we add wild type supernatant or supernatant from our exporter mutant, we have it supplements the growth quite a bit. If we take a step back and look at the petrobactin precursors, we find that spermidine does not enhance growth of petrobactin. Citrate does enhance growth of, enhance growth of petrobactin. That makes no sense. Growth of Bacillus anthracis in the absence of petrobactin. Citrate does. This is not unusual. It's been shown for E. coli, but it hasn't been shown for Bacillus anthracis, as far as I know. And the 3,4-DHB rings, which remember these have a high affinity for iron, especially when attached to the spirophore. Those enhance growth of petrobactin knockout quite a bit. So this tells us that, yeah, we have components in the supernatants that can facilitate growth of delta ASB. And at least 2 of the 3 petrobactin precursors can facilitate growth on their own. So what do the fragments look like? Based on our knowledge of the biosynthetic pathway, which I'm not going to show you, of petrobactin, I made some predictions for truncated biosynthetic products that might be exported into the supernatant and facilitate uptake of iron. So we have the standard 3,4-DHP spermidine citrate, and then we have pretty much every iteration of 3,4-DHP attached to a spermidine, spermidine attached to a citrate, and so on and so forth until you have 3,4-DHP with 2 spermidines and 1 citrate. So basically, 1 catechol shy of petrobactin. I then took advantage of individual knockouts in the biosynthetic operon of petrobactin. So I've been showing you thus far a full knockout, and here I'm going to show you strains that have individual knockouts of individual enzymes in the pathway. So I grew these in IBM, and I first needed to test that the biosynthetic products, the truncated products, were being exported. And so I did a catechol assay and found that all of the individual ASB biosynthesis mutants, except for ASBF, had catechols in the supernatant at least wild-type-like levels. ASBF had no catechols in the supernatant, which makes sense because, again, ASBF is responsible for the synthesis of the catechol rings. So I took these supernatants, fed them to the ASB, the full petrobactin knockout, and found that to some extent, all supernatants from all of the individual biosynthetic pathway mutants were capable of supplementing growth. So even citrate, potentially bound to spermidine, is capable of somehow coordinating iron and being imported into the anthracis cell, enough to increase growth at least a little bit. This isn't very helpful in terms of trying to identify which of the predicted fragments is actually binding the iron. Ashu and I did some experiments with using high-resolution LC-MS to try to identify which fragments are present in the supernatants of the export mutant, and we didn't have much success with that. But one reason for that might be if the petrobactin is being degraded as opposed to a biosynthetic truncated product, then it's difficult to predict where that enzyme might be cleaving and thus what those products are going to look like. And that's an important thing to know when you're using mass spec. Excuse me. Using mass spec to know exactly what your products look like. The ultimate question is whether or not this matters for disease. Okay, we've knocked out the petrobactin exporter. It still grows in IBM. Can it still cause disease in mice? So the answer to this question, I performed a mouse infection using our model of inhalation of anthrax, and found that within 5 days, all mice, whether they were infected with wild type or spores from either wild type or the petrobactin exporter knockout, 2407, all of them got sick enough that I had to euthanize them. So this suggests that at least under the conditions we use with the inoculum we use, that it is still possible for petrobactin components or these siderophore fragments to coordinate iron even in a host situation, and furthermore enable disease. So this gives me an updated model for how petrobactin is used by Bacillus anthracis. So it's exported through Apex transporter, recognized by this ATP type transporter system with redundancies. And then inside the cell, it's possible that it's cycled, but it's also possible that petrobactin is degraded in order to release the iron, and that the degradation of petrobactin creates fragments which are then exported again from the cell, gather more iron, and come back in. And data I didn't have time to show you today, there is evidence suggesting that there are fragments in the supernatants of wild type, and that these fragments use an alternate or different transport system. Okay, so I'm going to pause there for a second. This is a little difficult to transition into the next part of the talk. I do want to ask if there are any like super duper pressing, I didn't understand what you did in this scenario questions before I move on. If not, then we'll keep going. So the Bacillus anthracis lifestyle alternates between vegetative growth and dormant spores. So the spore is extremely durable, can withstand pretty harsh conditions for decades. And upon entry into less harsh conditions, small molecules, these are typically amino acids, can act to signal to the spore that it's time to move from the spore into vegetative growth through the process of germination. Germination is irreversible. Once it's triggered, it's done. So you have vegetative growth. The bacilli are really happy, and so they hit a high cell density, nutrients start to become scarce, and this triggers onset of sporulation, the process of moving back into the spore. Key to remember is that the spore is required for it's the infection particles, so you can't have an anthrax infection without first exposure to the spore, not vegetative bacilli. And next, that the bacilli are what's producing toxins actively growing and causing disease. And that in order for anthrax or anthracis to cause another anthrax infection, it has to successfully sporulate again for transmission. Just an idea of what the anthracis spore looks like. You have at the core, this is where the genome is packed in with chemical calcium dipolonic acid, and as well as your ribosomes and other transcriptional translational machinery. That's surrounded by an inner membrane, which is then surrounded by a modified layer of peptidoglycan called the cortex. That is surrounded by another membrane, the outer membrane. You have a layer of coat proteins, and then the exosporium, in the case of Bacillus anthracis. So my first question is whether or not petrobactin is important for germination. You never know where you're going to land as a spore, whether you're going to have adequate iron or not, or like not though. So this was where I started. We can visually track germination of anthracis spores because as they germinate, the core becomes hydrated, and this changes them from that phase bright spore that you saw in the very beginning to phase dark, and you get a corresponding drop in optical density. So a drop in optical density of around 50–60% correlates with 100% germination of spores present. And I found that the germination of the petrobactin mold is not really any different from germination of either wild type or bacillibactin. So the next step is looking at sporulation. And here I used a sporulation assay where I grew my cells in a specialized sporulation media over about 2 days. The time points at 12 hours, 36 hours, did serial dilution, and plated on BHI. One colony, one is presumably one bacilli, and then did a treatment at 65 degrees Celsius for 30 minutes. This inactivates the vegetative bacilli so that anything that grows after I plate those samples are spores. And this tells me the ratio of spores to total colony forming units in the culture. And something I mentioned to State a minute ago, why do I think iron is important for this process? In order to form efficient spores in the lab, you have to add crazy high amounts of iron to your sporulation medium. So this medium has 1.7 millimolar concentrations of iron, where, again, bacteria are trying to fight against 10 to the -18 millimolar for growth in the human host. So that's a pretty huge difference there. We found that petrobactin is required for rapid or efficient sporulation, even in this high iron sporulation medium. So here on the y-axis, I'm showing you either total colony forming units or the colony forming units of the spores at either 12 or 36 hours, and I have 4 different strains or conditions. Wild type, delta ASB with supplemented ferric-petrobactin and then bacillibactin knockout. And while total CFUs in sporulation medium don't appear to vary that much, when you look at the numbers of spores present, particularly, especially at the 12-hour time point, there's a pretty severe defect in sporulation by the petrobactin knockout. And that is complemented by the addition of purified petrobactin. So we seem to have a role for petrobactin in sporulation process. And I'm going to give you a little bit of background about sporulation. It is a highly regulated process that involves many gross changes to the cell structure. So in vegetative growth, it hits that high cell density, nutrient deprivation, and this triggers the first transcription factor in this cascade of transcription factors. SIG-H encodes or activates the enzymes and proteins required for the first step of sporulation, which is asymmetric cell division. So you have division of bacilli into pre-spore and the mother cell. And after this division, you then have compartmentalized activation of two different sigma factors, SIG-F and SIG-E, which enable the next step, engulfment, where the mother cell engulfs the pre-spore. You have a second round of transcription factors, which are again active in compartmentalized fashion, SIG-G and SIG-K, and from there you get building of the spore from both the inside out and the outside in. The cortex, the spore coat, maturation, lysis of the bacterial cell, and then you have spore ready for termination. I tell you all of this because when we look at the 500 base pairs upstream of ASB, the ASB operon, there are 3 predicted binding sites for sporulation-specific sigma factors. So just using Bacillus-specific prediction software. And I was curious, I mean, petrobactin is required for sporulation, but we don't necessarily have any evidence that petrobactin is being produced. It makes sense that it would be, but it's also low or high iron condition, so why would it be turned on? What would be turned on? So to try to explore this question a bit more, I created reporters. They're fluorescent reporters for the ASB operon, and I placed the 500 base pair promoter region along with a portion of ASB-A used to GFP, green fluorescent protein, and I did that immediately downstream of ASB on the genome to try to replicate the regulation as closely as possible. Here's what that data looks like. So what I've done is combined growth and fluorescence in the same chart for you. On the left, we have cell density. You can see we have a nice exponential phase in the sporulation medium, and then it plateaus. And along with that stationary phase and plateau, we see an increase in both transcription and translation of ASB. And this is consistent, this is representative of at least 3 different experiments that I performed with this. I confirmed this using fluorescence microscopy. So the microscopy was in part to confirm the fluorescence and also in part to try to tie the timing of ASB regulation or upregulation to sporulation. So what's the difference in time between when ASB is turned on and when you get the onset of phase right scores? So Zach helped me out with this. And you can see here at 8 hours, we have vegetative bacilli that are actively expressing ASBF. And this is the translational reporters. These are protein. At 10 hours, you can see the onset of phase right spores. So these are nearing the end of maturation. And we still see expression of ASB via our green fluorescent protein. Interestingly, the cells expressing GFP are not the ones undergoing sporulation. So ASB appears to be upregulated and then it shuts off at some point before we get phase-right spores. I think the timing of strata probably corresponds to around when you get activation of SIG-H and that asymmetric cell division. And Zach and I are working on trying to develop a movie so that we can track a few cells through the process of ASB upregulation into sporulation. The next question I wanted to ask is confirm is predicted binding sites. That doesn't mean necessarily that they are. What is activating expression of ASB? So to get at this question, I took advantage of a plasmid-based transcriptional work, again using GFP, that had only the 200 base pairs upstream of ASBA instead of the 500. And this region conveniently does not contain any of the predicted sporulation-specific sigma factor binding sites. So when I performed the same assay that I just showed you, growth and sporulation medium tracking growth and fluorescence, I found that while it was delayed, there is still a late-stage growth expression of ASB with this 200 promoter region as opposed to the 500 promoter region. So it doesn't seem that the predicted sigma factor binding sites are relevant for expression of ASB under these conditions. And this is a little bit confusing, because if they aren't responsible for its expression, then what is? Well, what I haven't shown you is that there are a lot of predicted binding sites upstream of ASB, and within that are regulators for ASB expression during vegetative growth, as well as during times of cell stress. So there are predicted binding sites for SIG-B, which is activated during it's a general stress response for Bacillus, as well as per-R, which is responsive to oxidative stress. These are both contained within that 200 base pair region that still allows for induction of ASB during sporulation. Well, previous work in the lab tells us that the ASB operon is induced and that petrobactin is exported during times of oxidative stress. So in low oxygen conditions, low iron, you have expression of petrobactin in the supernatant. If you add iron, you no longer have petrobactin supernatant. So okay, we have iron-dependent regulation. That's expected. Under conditions of high oxygen and thus high oxidative stress, low iron, okay, we have petrobactin. But even if you add iron, as long as there's oxidative stress, you still get expression of petrobactin supernatant, suggesting that there's a stress response that is overriding the availability of iron. Further work, well, this is actually back in 1987, a paper showed that in Bacillus subtilis, resistance to oxidative stress happens during stationary phase. So this is independent of the ability to sporulate. So regardless of whether the spore sporulates or not, it hits stationary phase and can no longer be killed by extremely high amounts, 10 millimolar hydrogen peroxide. And to sort of add some icing on top of the cake, we know that in using microarray of anthracis in our sporulation medium by previous graduate students, that oxidative stress response is upregulated during late-stage growth and moving into early sporulation. There are also enzymes involved in oxidative stress response incorporated into the spore itself. To sort of the last thing that I haven't really told you, seems petrobactin is important for sporulation. We have upregulation of the biosynthetic operon. We don't actually know, or I haven't actually shown you, whether or not petrobactin is present or being exported into the medium. So when I do catechol assays on sporulation media at 12 hours, I can't find catechols. However, Ashir and I have used LASY to do perform mass spec on the spores themselves, and the spores themselves contain or have petrobactin associated with them. We're collecting data now to try to figure out if using LASY to double check the catechol results, indicating that there's no petrobactin supernating. and also to check the cell pellet at that same time point. So this gives me a model for what I think is happening here. As cell density increases and nutrients decrease, the cells move from vegetative growth into a stationary phase. And accompanying this movement into stationary phase, you have oxidative stress conditions that upregulate HER-R, Sig-B, other stress responses. In turn, I predict that this stress response is upregulating biosynthesis of petrobactin, and that this petrobactin is protective against oxidative stress. I say that because in other siderophore systems, particularly those, actually only with those catechol-based siderophores, are protective against oxidative stress. And this is not due to iron-binding protein properties of the molecule itself. This is because there is some property inherent to the catechol rings that are protected against oxidative stress. And so the siderophore has to be present within the cell, not in the medium, in order for it to be protective. So that accounts for the absence of petrobactin from the supername and its eventual incorporation into the spore. So again, I told you before that sporulation is an important part of transmission. And I predict that if anthracis has inefficient sporulation, then that's probably going to have an impact on transmission. This is a really, really tricky thing to try to actually study in the lab. I think there might be some ethical issues trying to use mice for this kind of experiment because it requires that an animal bleed out, the blood be exposed to oxygen, that initiates sporulation of the bacilli, and then it hangs out and waits to be picked up by some other mammal. So my approach to this was to use aerated bovine blood, performing sporulation assays on those. I'm still collecting data, but it looks like petrobactin is required for full growth within bovine blood, even above heme and bacillibactin systems, and that you have a decrease in sporulation that is rescued by the addition of petrobactin. So close things out, wrap them up. I wanted to give you a big picture model of how petrobactin is relevant to anthracis disease, whether it be exported out from a cell or in spore biology. So we have infection of a mammal with spores containing petrobactin. Those spores are taken up by a macrophage, germinate in the lysosome, produce petrobactin in order to survive, and escape into the body of the macrophage where it replicates. Eventually escaping into the bloodstream, where again petrobactin seems to be important. We have growth to high density, death of the animal, exposure of that blood to oxygen, and a spike in oxidative stress. And production of petrobactin would then protect against oxidative stress to enable sporulation and for this cycle to continue. And with that, just a reminder of my model slides, and I am happy to take questions.
Thanks, Ada, for your lecture today, and frankly for everything from my heart that led up to your lecture today. Questions, please.
Hey, Ben, have you tested your AFC recorders and the antigen?
Yeah. Well, we did some preliminary studies, but we don't really have anything. But preliminary, yeah. Laura?
How can you exclude that iron is being depleted at the point? So for the second part of your story, like where you're talking about it's oxygen, low or high oxygen, but it's not the depletion of iron from the media or the blood that's also upregulating petrobactin expression.
So that's a good point. And I kind of skipped over that. In addition, so we already know that there's a role for oxidative stress and regulation of petrobactin. But to address your point that iron depletion is also an issue, yes, that is an issue. Bacillus, part of the reason sporulation mediums require such high amounts of iron is because the onset of sporulation and that transition requires high amounts of iron. So that connotation enzyme that I showed at the very beginning, that is huge amounts of iron during that transition process. So yes, there is a simultaneous depletion of iron and addition of oxidative stress.
Because I guess it could also just be the intracellular iron.
Yes.
Like it's not even necessarily the environment that's inside the cell.
Yeah.
Okay.
Regarding the first part of the talk, I was wondering if you were to compete in a 2704 mutant against the wild type cellular anthracis, either in vitro or in vivo, do you expect you would see any sort of effect? Partly because it just still surprises me that this is able to acquire enough iron to actually accomplish what it has.
Okay, so competing in wild type versus the export knockout, do I expect to see the effect? No. So that's the tricky thing about siderophores is they're kind of communal molecules since they're sent outside. So that would just further supplement the export. Now we do know that if you try to supplement an import mutant with petrobactin, they can't bring it in. Yeah.
So it's really curious to me, the first part of your story. So the wild type produces this siderophore petrobactin and can pull it back in. But then you also talked about these degraded petrobactin and how they're also able to supply the same sort of nutrients. Why would the wild type, I mean, to add more questions, why does the wild type care about that when it's got these intact molecules that it can bring in? Does the host routinely break down the petrobactin in some way, and that's why it has to be imported for these fragments?
Okay, so your question is sort of threefold. Yeah. All right. So you're asking why make the full length molecule with just the bacilli-laden, basically. Okay. So one is that our conditions are a little bit artificial. We're growing to extremely high densities in a closed system, and at least at the onset, if you think about a lone anthracis in a bloodstream, there's not going to be a whole lot of petrobactin out there. And so you're going to need the higher affinity that comes with the intact molecule as opposed to the fragments. That was part of it. The other part was, why bring them in? It's possible that we're using non-specific transporters for these. They're small enough. So they not require extra effort. Alice?
So what are the sporulation properties of the
I don't know. I haven't tested that. That is something I've thought about, but haven't had a chance to do it yet. A great question.
Yeah, is how some your, I guess I didn't necessarily see. So what was evidence that there were fragments going out? What could not just be those again?
Okay, so bacillibactin has catechol rings. So is it possible that we were detecting bacillibactin, the supernatant, and that it's not petrobactin components? And I have a twofold response to that. One is that the time points we were looking at are within 6 hours of growth in this medium. Bacillibactin isn't seen in supernatants until 20 hours or after. So the early exponential phase of growth is petrobactin. The second is that Ashu and I confirmed the lazy data with LCMS, and we weren't able to find bacillibactin present in the samples. So it doesn't look like bacillibactin is being exported, or at least it's not being exported.
So now having this apex mutants and the releasing technology, can you ask other questions? What other things might be transported by apex besides petrobactin?
Yeah, that's an interesting question. So something that Ashu and I dabbled with, but it turned out to be a lot more complex than the time return warranted, is with lazy, because you can look at so many different molecules at the same time, you can sort of get like a thumbprint of what the supernatant from each mutant looks like. And then theoretically in the future, you could compare those thumbprints and try to identify molecules that are being transported or not being transported based on the mutant you're looking at. I think that's a possibility. It would take a lot of work to figure it out. Well,
A couple more, yeah. We're past our top of the hour.
So why do you think that method has so much iron to get to use sporulation, whereas in nature there's such a low concentration of it? Do you think there's other anthracis and other metals that are capable of complementing this?
okay, so why do you have to add so much iron to the media in the lab when there's not that much iron present in wild? So when bacillus anthracis reach the point of entering into sporulation, there are actually two different pathways. They can either sporulate or they can turn on complements and hope to take up DNA that will further enable their survival in these conditions. So really only a small portion of your population in the wild is going through sporulation. The second part of that is in the lab, the sporulation media, it's optimized to get as many spores as possible. So we're trying to really shove everything in the direction of spores as opposed to complements. And as for other ionophores, I mean, cobalt can sometimes, in like lactobacilli, they use cobalt instead of iron. So it's possible, but it's not really very efficient.
Thanks.
Molly,
Sorry. Always has the last word.
To summarize your findings, based on the in vivo test, what would you conclude about the role of ferric-petrobactin versus the category of products? It's an essential factor to grow.
Okay, so when you say other metabolic products, do you mean other types of iron acquisition or relative to other like clearance factors?
If I understood you, what you're saying that in the apex, you're thinking of exporting ferric-petrobactin, but you might be exporting.
Right.
So that appears to be sufficient in vivo.
Yes. Yes.
So in summary, we think that the bacterium is sexual.
You mean the intact versus the fragments? Okay. I think yes, because there is a difference in how tightly petrobactin binds iron and thus its ability to scavenge iron efficiently, as opposed to the petrobactin components. So I didn't show it today, but I've done experiments titrating dipyryl, which is an iron chelator, and the exporter mutant is inhibited, its growth is inhibited before the wild type at a certain concentration of dipyryl. So in those titrations, petrobactin has a stronger affinity for iron. So I do think it's important.
Thanks, Sadie, again. Thank you all. Committee, we're going to the fifth floor, pretty much sure we'll find it. Fifth floor, your department. Probably.